Hello, so we are ready for our 7.2 lecture. This deals with eukaryotic cells. If you remember back to last time, we were talking about cell theory and the idea that all living things are made of cells, cells come from cells, and any sort of structural component of any organism is built from cells. So collectively, we've got a lot of cells. So let's take a look at the general structure of a eukaryotic cell. Now that's opposed to a prokaryotic cell, which would be something like bacteria. We're following along with our lecture slides, so if you want to follow along with that, that would be pretty useful. So let's talk a little bit about a eukaryotic organism. So if you're a eukaryote, we said that you were more complex, you had a nucleus, or you kept your DNA in a nucleus in a central region. You had more complex structures, you're larger, and you can work together to be multicellular. So why can it do this? It's because it's just overall a more complex structure, and it can work with other cells. It can specialize better, it has better cellular machinery. And that's why you get to build like really cool stuff like budding rabbits and people out of eukaryotic cells. And the only thing that's prokaryotic is bacteria. So that's really good. So in a eukaryotic cell, those little structures that are inside of them, organelles, which means literally little organs, it has little structures that do a particular job, and they work together to carry out the cell function. This is the analogy that's most often used, is that all these organelles and the cell as a whole is like a factory, and you've got different parts of the factory that do different parts of the manufacturing process to keep the factory running. So we'll use that comparison often as we go through this. Whenever we first start looking at a cell, I need to point out, first off, all the cells that you're going to see and most of the diagrams that you'll see in the slides and the models that we'll make and all this other things, they will be rather generalized, generic sort of cells. They kind of are round and they kind of have a big fat nucleus, and that's about it. Those are really rather generic. They're not real specific as to a particular type of cell for a human. For example, a muscle cell is rather long, and it's multinucleated, many nuclei. Nerve cells are highly branched with one nucleus, and they can be rather long. Cells in your receptor cells in your eye, or cells that I guess that are part of your skin, those are flat. So there's all these different types of cells, and so whenever we start drawing or talking about cells, realize that we're just talking about a really generic cell. Now all these other cells have these same structures and they still do the same things. It's just that we're not looking at one specific type of cell. We're looking at like the average cell, which necessarily isn't that common, because all your cells are doing a job for you somewhere, so they've become specialized to do that job. When we start looking at a cell, our generic cell, okay, we are going to, let's just make a big circle. All right, there's our cell. And usually on the inside they show something like this, all right? So let's just kind of start here. This outside barrier would be the cell membrane. Inside here, we have this region called the nucleus. It's got its own membrane around it, just like the cell has a membrane. It's separated from the rest of the inside of the cell. As all of your membrane-bound organelles will have their own little membrane around them. That's good, and we'll talk about why. So you keep your DNA here in the nucleus. Now the rest of this stuff, okay? This rest of this area collectively is all just called the cytoplasm. Okay? That plasm that's inside of this cell, the stuff that's in the cell. This prefix, this word part cyto, means cell. So if you hear the term cytoskeleton, it means your cell's skeleton. You have those. Okay? It's not made out of bone, it's made out of protein fibers. Right? So anytime you see that word cyte or cyto, it means cell. So this is your cytoplasm in here, all this stuff in there. And then inside of that, you've got the fluid part is called the cytosol. This is fluid. So think about this, your cytosol, your cell's solvent or cell solution, right? You've got water with different things dissolved in that water. It's holding on to it, so that way whenever you need some material, the cytosol has those materials dissolved in it, whether it's sugar or oxygen or whatever. It's like, I don't want to say soup, but it's kind of like a soup that has everything like dissolved in it, and it's ready for all these different parts to use. Okay, the nucleus itself, it contains your cell's DNA, right? So we compare this to a control center of a factory. I'm going to maybe specify it's more like it doesn't make decisions. What it does is it's just like code. Your DNA is a code, right? Your genetic code. And this is like the library that houses all of the code. And if you had a big giant factory that's really super complex and you had one copy of how to run every single bit of machinery in that factory, would you just leave that instruction manual lying around wherever and carry it all over the place? You wouldn't. You keep it locked up in a central region where everyone else can access that information, and you would guard that pretty close. That's what the nucleus does. It has a nuclear envelope or your nuclear membrane that surrounds that and keeps the DNA safe, so that way it doesn't get damaged or lost or whatever it might happen to that DNA. And then it can hand out little bits at a time. Would you like to know how to do this? Okay, I'm going to make a copy of it. I'm going to give you the copy, and I will keep the actual DNA myself. And so that's how that works, right? Whenever these little, the strands of DNA that we talk about, that is called, chromosomes. That's one single long strand, and that contains segments that control traits called genes, right? So you talk about genetic traits or whatever. Those are controlled on segments of DNA, and we keep it all here. It tells you how to build stuff. All right, so kind of on that vein, what if you do want to build something, okay? You need some way to take that DNA and then actually manufacture something out of it, right? One of the primary non-membrane bound organelles is something called a ribosome, and they're usually just drawn with a little dot. They're not real big, right? I'm just gonna put a couple little dots. Wow, that's real good drawing. And these then would be ribosomes. Now the important thing you need to remember about all these ribosomes, and there should be like hundreds more in here, I've only drawn 3, is that ribosomes take genetic material from the nucleus and they read it and they actually do what it says to do. So if it says build this type of protein, they read it and they build that type of protein. They grab amino acids from the cytosol, right, those monomers of proteins, and they start assembling them. They start hooking them together. That's their job. They just assemble proteins from DNA instructions. And that then allows you to build everything else. Prokaryotes like bacteria have ribosomes as well. They're that important. It's really important to be able to manufacture proteins. And so you get some messenger RNA from the nucleus that tells it which proteins to assemble. So that is a site of protein synthesis. Another structure that's or structure that's pretty predominant is something called a endoplasmic reticulum. And it tends to be close around the nucleus. Okay? So instead of saying endoplasmic reticulum like 15 times, we're just going to say the ER for endoplasmic reticulum. What does that word mean? Endo means inside the plasm, inside the cytoplasm. So endoplasmic. Reticulated means it goes back and forth. So it's this thing in the cytoplasm that goes back and forth. It's a big, giant folded membrane, right? Remember we talked about membrane, we mentioned membrane-bound organelles. Well, it's a big folded membrane. So let's go back to this, why do we need membranes around everything? Well, let's say we have a factory, and you make pens. And let's say you have a machine that just makes pens. Makes a pen, drops it out. Makes a pen, drop it out. Or markers or something like that. So are you just going to have a bit of machinery that's making this marker and just drops it out, and you're just going to have a big pile of loose markers or pens on the floor? No, you're not going to do that. You're going to have a bin underneath that machine that catches that particular product. let's say. So very much like that, your endoplasmic reticulum with all of its folded membranes, it acts as a big giant container. So that way, as you start making things, you can hold them in place or separate them from the rest of the stuff being made in other places. You don't want to drop your pens and your markers and your pencils and your erasers all in one big pile on the floor. You want to keep them separated. So just like that, these membranes allow organelles to separate their cellular processes and the materials they make from each other and keep that separate from the rest of the cytoplasm. If you were making something really super delicate, like the inside of a watch or something like that, you wouldn't just have it open on the floor with all the other dust and everybody walking by and everything and then make one and just toss it on the floor. No, you'd isolate it so you didn't get anything of the components dirty, you'd make it really nice, and then you'd package it nicely, right? And that's what membranes allow you to do, right? The ER comes in two different flavors, smooth and rough. The rough ER is studded with ribosomes. I told you there was more. And those ribosomes are embedded in the membrane. So this part would be called a rough endoplasmic reticulum. This is be a smooth endoplasmic reticulum. So the rough has a bunch of ribosomes. What does it do? Those ribosomes make proteins. The ER then allows it to compartmentalize or package those proteins or hold them in like a storage tank until they're ready to do something more with them, separating them from the rest of the particular part of the cell. Once you're ready to do something with that, there is another folded membrane structure that looks a little different called the Golgi apparatus, and it tends to not be around the nucleus, and it kind of looks like this. I'm just going to put Golgi. Sometimes it's called the Golgi body. So this is an interesting one. Once again, it's a bunch of folded membranes. But the Golgi apparatus or Golgi body functions like the FedEx for your cell. It gets little membrane-bound transport vesicles from the ER, and it accepts those. So, like if you had a factory and had that big bin full of markers and you wanted to I need to go ship these out to different places, you'd move them somewhere, they'd get sorted out, packaged, and then they'd get sent out in their own nice boxes. Okay, that's what the Golgi apparatus does. It receives things from the endoplasmic reticulum, it further processes them, and then it exports them from the cell. So if you were going to make some saliva, because you do that every once in a while, you've got enzymes in there. The enzymes, the specialized proteins, are made here. Once they're made here, they're packaged in a little membrane package that's shipped to the Golgi apparatus. The Golgi apparatus fully modifies that, gets it ready for export, and then it pushes that out of the cell where those little membrane-bound chambers fuse with the cell membrane, and it dumps something more like spit or salivary amylase in this case into your salivary glands so you can go eat something. All right, so you've got cells doing that right now, and they're using that Golgi apparatus to package and get that material ready to be sent out. Lysosomes are specialized little membrane-bound structures. Boy, everything is surrounded by a membrane. Well, that's so they can do specialized things instead of just having everything all mixing together. Okay? Lysosomes are small structures that are filled with digestive enzymes. I'm going to make them green. Make them green. Okay? So that word lyse at the front of lysosome means to split or to break up. So lysosomes are little small membrane-bound chambers full of digestive enzymes. So let's say one of our ribosomes isn't working anymore. What are we going to do? How are we going to repair that cellular machinery? Well, maybe we won't. Maybe we'll just break it totally apart and make a new one. In that case, the lysosome can fuse with it and dump a bunch of digestive enzymes on it and disassemble that particular piece of cellular machinery. Remember monomers and polymers, it will depolymerize it. You don't want, it's like the cell's cleanup crew. If they're a factory, it's like the custodians / janitors. And if some piece of machinery is no longer functioning, they'll tear it apart and build, and then someone else will come along and build a new one there. Right? That's what lysosomes do in your cell. Mitochondria. Okay? Now, everybody's going to want to say the mitochondria is the powerhouse of the cell, and I don't like that because it's not specific. Maybe we'll talk about some better ways of that. You guys have all seen tons of diagrams. Mine doesn't need to be that special. So mitochondria are the site of cellular respiration, and that's what I really want you to know. And cellular respiration is how you generate ATP. Usable cellular energy comes in one particular form, ATP, which is usable cellular energy. It's short for adenosine triphosphate. That is a chemical that you use as like a cellular energy battery. You constantly got to keep making it to keep your cell alive. And that happens in your mitochondria. If your cells have a higher energy requirement, they need more mitochondria to provide that. So like the cells in your cartilage in your ear, like how much do you do with that? Not much. There's not going to be a lot of mitochondria there. However, some sort of large muscle cell or in your liver or heart, you're going to have a lot of mitochondria because you need to generate lots of ATP for those cells to be able to function, right? So that's what your mitochondria is. They're the power plant, if you will, okay, but what you really need to know is that that's the site of cellular respiration to generate ATP, right? You've got to have ATP or you don't get to live anymore. So it's a pretty good thing. Now, if this was an animal cell, or this is an animal cell at this point, because I don't have a cell wall and I do not have chloroplasts. So if it was a plant, I would draw an additional layer around there of the cell wall, and I would add some structures, some green structures, because they'd have green pigments. That would be the chloroplasts. Chloroplasts allow you to turn sunlight, light energy, into carbohydrates. You capture that and you rearrange some things, and you actually end up making sugars, so that way you can survive. Plant cells don't get to do that. Sometimes plants will have a large storage chamber called a vacuole. Animal cells tend to not have big vacuoles. Instead, we just use lysosomes to store things. The last thing that this has that I'm not going to draw is a cytoskeleton. Remember we said that word part, cyto means cell. So your cytoskeleton is your cell skeleton. It's really just a network of protein fibers. So it's really tempting. It's really tempting to be like, okay, this is a bowl. This is like the soup in the bowl, and I've got a giant like meatball in the middle of my big bowl of soup, and it's all sloshy and everything. No, it's not at all like that. This is just a diagram. It's a model. It's not really like the real thing. Okay, this has a network of protein fibers to it that helps to anchor the organelles in place so they aren't like sloshing around every time you walk, and it helps to hold the cell membrane to allow it to keep its shape. Also, there's proteins that can transport materials along that cytoskeleton. The cytoskeleton also aids when the cell is ready to divide and pull apart and make two cells. It helps with that as well. So the cytoskeleton is really important. We never ever draw it, hardly, because if we really did, there would be a bunch of marks all over the place as we tried to draw those protein fibers. Think of it more like this. If you've ever seen a big like circus tent or a large tent for a party or a reception, it's got all these metal tubes with some cables going through it, and then you've got all this room on the inside. Think of that like the cytoskeleton inside of a cell. It's what supports and anchors things in place. Okay, we'll stop there and we'll pick back up with cell boundaries next time, and we'll talk a lot about the cell membrane.